He's on his way. It's my pleasure to welcome Peter Dervan to the podium. Peter will be talking about targeting transcription factor DNA interfaces with small molecules, an area in which his career has been spent. He's really spent his whole career at Caltech, or at least his academic career, professorial career, and is highly recognized, as you all know, most recently with the National Medal of Science. So Peter, it's a great pleasure to have you here.
Okay, thanks. Well, thanks very much to the organizers, particularly Steenbock Symposium. I'm grateful to participate in this fantastic group of speakers here at Madison. I spent 2 years of my life here at Madison. Most people don't know that, although I got my PhD at Yale. 2 wonderful years at Madison. It was a lot colder then, back in the late 67 to 69. You know, when people come back to celebrate the life and achievements of a great person in science that's inspired all of us, Govind Karan is one of those people. It must be a real joy for the students to come back and see each other. Talking to Marge last night at the Madison Club about this. So I wasn't a student of Govind, but in a way I'm still part of the DNA Club and the Nucleic Acids Club, and that he was an inspiration to me. And I tried to remember, was there a time in my life, other than being inspired by his work in the literature, that I remember meeting him. And I did, and it's crystal clear. We all go to different meetings and to different universities and meet people, but maybe we walk away and 20 years later can't remember that meeting. And I remember with great clarity that in 1987, we didn't have word processors then. This was typed clearly. I was visiting the department at MIT. I think I was a T.Y. Shen visiting professor or something. And there on my schedule at 10 o'clock was an hour to be spent with Govind. So he was, this is 87, so you can do the math. This is, you call that now the middle of his career, I guess, for us old guys. And I remember we sat on the floor together, and he put a kettle on, and then we spent an hour talking about his efforts to synthesize nucleic acids. And I just want to make 2 comments, and then I'll get on with some chemistry, my own chemistry. And, you know, back when he first started to figure out how to make a phosphodiester bond, dinucleotides and trinucleotides, mainstream synthetic organic chemistry didn't think this was very interesting. So he was on the outside looking in. This wasn't fashionable organic chemistry. After all, these were boring polymers. And then, even for the biologists, before you get to self-satisfied, biology didn't think this was ever going to come to any good. It's too slow. Teachers, how are you going to chromatograph these things? And then, of course, with your students' work, Mark Brothers in particular, this was reduced to perfect chemistry that we now take for granted in machine-assisted synthesis. So what's the lesson for me and for all of us? That don't worry about being on the outside, and to have a vision for a hard problem, and maybe someday it will happen. So what an inspirational person is Govind Karan, and I'm excited to be here to help celebrate his career in life. Now, I'm a little bit nervous about giving a 15-minute talk because I think that's all I have left. And so I probably did what the other speakers did. I took my standard 60-minute talk and I threw out 3/4 of the lecture. So I'm not sure what's on the UBS drive. What did I throw out and what didn't I? But this story is really a simple story, and I could probably do a chalk talk. And that there's really just, what's the question? You know, what do we know? And then importantly, and I'd like to end early enough to get some feedback from this heavily biology audience, what do we have to worry about going forward? And so we all know that the cell senses its environment, growth factors, small molecules, even by oxygen concentration on the surface, and then to some signal transduction pathways, it sets off gene expression programs. And a key player in the cell creating these gene expression programs are, of course, the protein-DNA interactions, transcription factor DNA interfaces. And so single transcription factors can turn on hundreds and hundreds of genes, program networks. And so the question that we've been asking in our research group is in these interfaces where information from the outside is converted into gene expression programs, could small organic molecules that are cell-permeable perturb this protein-DNA interface in a predictable manner and alter gene expression pathways? The reason this might be practically useful someday, and this has been recognized in the biology community, not necessarily just Jim Darnell, but we know that in cancers, human cancers, that many of the human cancers have now been characterized as overabundant or overactive transcription factors. So antigen receptor is a classic example. Overabundant or overactive antigen receptor creates the condition of prostate cancer. And even from metastasis, hypoxia-inducible factor and the low dioxin concentrations turns on again hundreds of genes in order to achieve vascularization and metastatic state. Okay. Now, in this review by Jim Darnell, he was really thinking about chemists, organic chemists, Stu Schreiber, like screening libraries of compounds to target the proteins themselves. And in our research group, fascinated with nucleic acids, we wanted to ask the question whether we could build small cell-permeable organic molecules that could, in every case, disrupt these different protein-DNA interfaces that have been characterized now in the field of structural biology and physical chemistry since 1987. So nature, of course, uses a diverse repertoire of protein folds to bind different sequences of DNA, some in the major groove, some in the minor groove, and some in the major and minor groove. And so this is an inspirational graphic that I took from a recent paper by Steve Harrison and Tom Maniatis. It shows 55 base pairs of DNA in the interferon beta enhanceosome. It shows an ensemble of proteins. This is a composite of crystal structures. Most of this cooperativity is through the DNA, but it reminds me to tell you something you already know, and that is most of these transcription factors, 1, 2, 3, 4, 5, have binding site sizes of 4 to 6 base pairs. So this is not an siRNA exercise, an antisense exercise. That we don't, in fact, want molecules that bind larger than the 4 base pairs. We're going to create molecules that are anti-transcription factor in their activity, and we bind computatively to 8,000 sites, the same 8,000 sites that, say, an antigen receptor would bind. So the goal in our program, the progress report this morning, is to build organic chemicals, organic molecules that integrate 4 properties. So in a single molecule, we can't sacrifice one of these properties. That is, they have to bind DNA comparable to DNA binding proteins. They have to be able to access condensed DNA. That's a key issue in the cell. They have to somehow, when they bind the DNA, disrupt the protein-DNA interaction in all cases. And of course, they have to be small enough to be cell-permeable without the use of transduction agents and other means. So the chemistry that we've come up with, and you've all seen these papers in literature, this is a minor groove DNA recognition code. And so we look at the edge of the Watson-Crick base pair, so the DNA is intact, a double helix. And we can think of an AT base pair, or TA, as having a hole at the edge of the minor groove, and a GC is having a bump. And so this is really just bumps and holes like Braille. And you can imagine that since all the information that is contained in the human genome and makes us who we are is in these 4 base pairs, then we're going to create organic molecules that, rather than being solidated by water, will be specifically solidated by these specific edges on the floor of the minor groove. So we took as our lead molecule, this is really an exercise in natural product chemistry. There's a natural product called isomycin. These are N-methylpyrrole amino acids, 5-member ring amino acid, again amino acid, taken 3 times. These have been shown by the biophysical chemists in the 70s as binding AT-rich sequences, real pioneers in those days. And then with the importance of structural biology, both from crystal structures and NMR, Dickerson and later Wemmer showed that these crescent-shaped molecules could bind in 2 stoichiometries, 1 to 1 and 2 to 1 depending what the AT sequence was. So there's some complexity with regard to stoichiometry. Now the reason these bind to AT sequences is the CH at the corner of this crescent-shaped molecule is sterically permissive for the whole, and a CH, this molecule, these rings, is sterically not permissive for a GC-based pair. And so to fast forward 10 years of organic chemistry, I'm just going to skip it one slide, tell you that using organic synthesis, one can create then 2 new rings, an indazole, that is a nitrogen lone pair in the corner, an unsymmetrical ring pair, an indazole paired with PY binds a GC-based pair and distinguishes it from AT and CG. And then by putting a bump on the ring instead of a hole, then the hydroxyl pyrrole paired with PY can fit into the hole here, and an HP-PY pair distinguishes TA from AT. So 2 new rings paired 3 different ways, then bind the 4 Watson-Crick-based pairs. And this is not a unique solution just for this sequence. You'll see that these rings are modular. And so this should work for any sequence of nucleic acid on planet Earth, whether it's tomato DNA, yeast DNA, or human DNA. That is, once you know the DNA sequence, the graduate student would know what ring pairs to synthesize in the laboratory. And so this is a crystal structure from Doug Reese's group at Caltech in '98. And you can see that this is a lovely example of what chemists in the field of molecular recognition would call shape selective recognition. These rings, the 3 rings paired 4 ways, this is a crystal structure, the stacked rings side by side, fit the shape of canonical B-form DNA. Now in this structure, there's only about a 1 angstrom widening of the minor groove. And so in this particular case, there's not too much structural change in the nucleic acid structure. Now, in practice, we connect the carboxy end with the amino end with the 3-carbon link. We call this a hairpin PY polyamide, and we refer to the unsymmetrical ring pairs as pairing roles. And quite often we'll just use a simple nomenclature, ball and stick, for the hairpin to indicate, if you read the sequence, you'd know what a series of molecules to synthesize. And we synthesize these by solid phase methods in rapid fashion in high purity. Now, over time, with these 6-ring hairpins, the central ring pairs code for 4 base pairs. The turn codes for an AT or TA for steric reasons. It doesn't like that, it's a cyclic bond. And whatever's on the tail also codes for TA or an AT. And so this would be a 6-base pair binding well an 8-ring hairpin. Now, over time, we've synthesized lots of these by solid phase methods. And here's a small library that we've put together in one paper, so there's one-stop shopping. And you see that the binding affinities characterized by quantitative footprint titration are in the range of 10 to the 9th to 10 to the 10th. So they have the affinity of a protein, and for a single base mismatch, the affinity would drop off by a factor of 5 to 10, not unlike a protein. Now, in practice, in collaboration with Joel Godesfeld at Scripps, we looked at a lot of protein DNA complexes, and by gel shift asked the question whether these sequence-specific programmable hairpins could displace the protein. And almost in every case, it always worked. And I never understood that. Trivially, if it was in the mining group, that's a steric blockade. But why should a molecule binding in the mining group displace a protein in the major group? And I never got why, in most cases, this always seemed to displace any transcription factor that we had. And I have a graduate student in my group, Dave Chenoweth, who really had a passion to ask the question, what does that term look like? Because after all, the Reese crystal structure was the unlinked material. And so he was able to co-crystallize this cyclic polyamide, a cycle for reasons of symmetry. We were able to get a 1 angstrom crystal structure of this cyclic polyamide bound to a short piece of DNA. And so here we see the turn on the floor of the mining group, which was the original goal of this project. And we can see this methylene pucker in the beta position down, and we quite often have a carbolamine at that position. All well and good. But when I saw this structure, and this just shows you a nice example of the hydrogen bonds, the edges of the unsymmetrical pair, literally reading specifically the edge of the Watson-Crick GC base pair. Okay. But the reason I'm excited to show this structure today here in Madison is that when I saw this structure, and David also solved the high resolution the naked DNA, the unbound DNA, you can see clearly, without getting your measuring stick out, that the minor groove of this DNA bound with the cyclic polyamide in the minor groove has been widened by about 4 angstroms. So effectively what these hairpin polyamides are is they're not only programmable sequence-specific binding molecules, they're allosteric effectors for the DNA. So what happens is in the native DNA, the minor groove width is this, and then the minor groove is widened by 4 angstroms. And of course the major groove then is compressed. So the major groove is compressed by 4 angstroms at that sequence, and so all the contacts for any given transcription factor have just been obliterated. So we have basically here sequence-specific allosteric effectors for DNA. I've got about 5 minutes left, so let's say, what do they do? Well, I think the 2 key issues is can they get into a cell, and then importantly, can they access chromatin? And so we know that they can at least bind to chromatin formally. We, of course, have the work of Tim Richmond since the late 1990s. And Karen Luger at Colorado State was successful in crystallizing the hairpin polyamides with the nucleosome core particle. In fact, we now understand why these crystal structures are so easy to come by. It turns out that the grooves, the DNA 146 base pairs wrapped on the protein core, the grooves are opened by 2 angstroms. And so it turns out the polyamides all set up at once to open the minor groove. So the wide minor groove of chromatin is in fact the perfect place for these sequence-specific hairpins. These are separated. This is an inverted repeat, 74 base pairs, and so these are 80 base pairs apart, but only 12 angstroms apart in the 3-dimensional space on the particle. Kind of cool. Now the question is, are they cell permeable? So by putting fluorescein on the C-terminal end, we're able to show that in HeLa cells at micromolar concentrations, this is a FITSI labeled polyamide. In unfixed cells, it's very important not to fix the cells, that at micromolar concentrations, these alien hairpins will be taken up by cells. So let me just in 60 seconds, 120 seconds, give you a sense of what the data looks like when you do a cell culture experiment. Now we chose for our first couple of cell culture experiments to pick an inducible system. The hope was that an inducible system, the promoter might be accessible and we can induce and then compete with the transcription factor. So the 2 systems we looked at, this hypoxia-inducible factor and antigen receptor. I'll just show you the hypoxia-inducible factor this morning given the time constraints. Now HIF is a